guys, welcome to Bio 112 Online, your home for biology education. Today's topic is the respiratory system and gas exchange. From our last video, you should remember that when we were talking about the respiratory system, in this case, we're talking about the mammalian lungs. These mammalian lungs will draw air from the environment and will be used to exchange oxygen from the atmosphere being pulled into the body and carbon dioxide being produced within the body being sent out into the atmosphere. This is being controlled by a coordinated effort between your diaphragm, which is going to be this sheet of skeletal muscle at the base of your rib cage, in coordination with the other muscles of your rib cage. When they contract, they will increase the volume of your thoracic cavity. In doing so, they will increase the potential volume of your lungs, allowing you via negative pressure to draw air into the lungs. When they relax, they decrease the volume of the thoracic cavity, allowing you to push that air back out again. We refer to this as tidal breathing. Inside of your lungs, you have respiratory surfaces associated with the alveoli. These alveoli are going to be small dead-end branches and sacs at the end of the bronchioles. These alveoli will take oxygen from the air and allow it to dissolve into water. As it dissolves into water, it will then have to diffuse across the boundary layer of epithelial cells into the tissue, and then subsequently diffuse into the capillaries. Recall the main drivers of this process are first going to be Henry's law, which is going to describe the solubility of gases within a solution. Within Henry's law, the solubility of the gas, or the concentration at saturation of the gas, is going to be proportional to the partial pressure of that gas. Within the field of physiology, we simply use partial pressure of the gas as a surrogate or as a unit of measure of the concentration of that gas. And so as we go forward through today, when we describe how much oxygen is in solution, we'll be referring to it as a partial pressure. When we're doing this, this is going to be a shorthand for concentration of oxygen in solution. Once we have that concentration of oxygen in solution, we then have to rely on Fick's law to describe the movement of that gas from the water in the alveoli into the tissues of the lungs and subsequently into the capillaries within the lungs. Fick's law states that the rate of diffusion will be equal to a constant times the surface area over which gas exchange is taking place times the difference in concentration between where the gas is coming from to where it's going to, and finally divided by the thickness of the gas exchange surface. The lungs are set to specialize to maximize the rate of exchange by, one, having a very large internal surface area within the alveoli, cumulatively. Second, by maximizing the difference in concentrations between the capillaries and the alveoli to ensure a fast rate of exchange. And finally, to have the thinnest possible thickness over which that gas exchange needs to take place. As a result, the capillaries are quite close to the surface of that gas exchange. So if we go through and we put this in the context of the circulatory system within the pulmonary circuits and take a look at what's going on in those capillaries. As the blood arrives from the heart in those capillaries, it will have a lower partial pressure of oxygen and a higher partial pressure of carbon dioxide in the blood relative to the air. As a result, because the partial pressure in the air and that thin layer of water in the alveoli is higher than the partial pressure in the blood, oxygen will diffuse into the blood. Similarly, because the concentration of carbon dioxide or the partial pressure of carbon dioxide is higher in the blood than it is in the water that lines the alveoli or in the air within the alveoli, then carbon dioxide will diffuse out of the blood and into that air. Now we run into one problem. This problem is that the concentration of oxygen in the fluid of the plasma of the blood can't get very large. This is because, as we said, the amount of oxygen that water can hold is relatively small, only 1/1000th of what is when the concentration in the atmosphere. And that's at very cold temperatures and at pure salinities. The water within your blood is actually quite salty, which will further reduce the solubility of oxygen. In addition, your body temperature is quite warm, further reducing the solubility of that oxygen. As a result, if we were to rely only on the plasma of the blood, we would not be able to transport enough oxygen to the tissues that need it. As a result, the body relies on a specialized subset of cells to transport that oxygen, specifically the red blood cells, otherwise known as the erythrocytes. Erythrocytes are non-nucleated blood cells containing the respiratory pigment called hemoglobin. When I say respiratory pigment, what I'm referring to is a protein that stores and or transports oxygen. Hemoglobin specifically is an iron-based oxygen-binding protein found in vertebrates. It has 4 polypeptide subunits, each of which contains a hemigroup with an iron atom at the center. The iron atom is specifically what helps the hemoglobin store oxygen. The dissociation curve is going to be the relationship between oxygen concentration in the blood plasma and the oxygen that is bound to the hemoglobin. This dissociation curve is an S-shaped curve due to the cooperativity between binding sites in the hemoglobin. Specifically for cooperativity, it is a process in which the binding sites of the hemoglobin molecule become more available to oxygen if some of the other sites on that same molecule are already occupied by oxygen. As a result, if one site gets bound to oxygen, then the other sites bind that oxygen slightly easier. They become more available. If a second site gets bound, then you have an even greater availability. If a third site, then even greater availability. The binding of oxygen to hemoglobin is reversible, though. So oxygen can diffuse on, bind to the site, and then re-release itself. And its ability to bind to that site is going to be dependent on that cooperativity. Binding sites of one subunit changes the conformation for the other subunits, increasing the affinity. But then the reverse is true. If one oxygen leaves from one of the 4 binding sites, then it makes it more likely that the others will also leave. Different forms of hemoglobin can have different dissociation curves and therefore different affinities towards oxygen. This becomes especially important in the transfer of oxygen between mother and babies that are still in the womb. Fetal hemoglobin has a dissociation curve shifted to the left relative to adult hemoglobin. As a result, for the same partial pressure of oxygen, the fetal hemoglobin will be more likely to bind the oxygen. As the oxygen gets released from the adult hemoglobin, it will preferentially get bound to the fetal hemoglobin, transferring it from mother to baby because of this difference. Another aspect of that hemoglobin is that it does not maintain that dissociation curve under all conditions. Very importantly, there is what we refer to as the Bohr shift, or a lowering of the affinity of the hemoglobin for oxygen caused by a decrease in pH. So why is the blood going to decrease in pH? This happens because in areas that are producing large amounts of carbon dioxide due to cellular respiration, then that carbon dioxide will form carbonic acid. The Bohr shift in response to lower pH helps release more oxygen from hemoglobin in metabolically active tissues. These tissues that produce more carbon dioxide will therefore have a lower pH in the capillaries due to that formation of carbonic acid. So let's consider how this oxygen then travels in the blood. Starting off in the alveoli, you have oxygen dissolving into solution, according to Henry's law, and diffusion into the capillaries. Once it gets into the capillaries, you have oxygen interacting with the red blood cell, entering onto hemoglobin. As a result, the partial pressure of oxygen increases within the capillaries of the alveoli, and therefore you have oxygen becoming more and more bound to the hemoglobin. As more oxygen is bound, it lowers the partial pressure slightly, meaning that you have a greater diffusion. That blood is then transported from the lungs to the pulmonary veins. From the pulmonary veins, it will then enter the left atrium into the left ventricle, entering then the aorta, going either to the anterior or posterior systemic circuits. As it does so, that blood will arrive in the systemic capillaries. At that point, the blood will have a higher partial pressure of oxygen relative to the interstitial fluid, and it will have a lower partial pressure of carbon dioxide relative to the interstitial fluid. As a result, oxygen will therefore diffuse out of the blood and carbon dioxide will diffuse into the blood, according to Fick's law. However, you still have most of the oxygen actually stored on those erythrocytes. So at that point, as you have the initial diffusion of oxygen from the capillaries into the active tissues, you have two main things going on. First of which is you have that Bohr shift, which means that the affinity for oxygen is lower. As you have more oxygen diffuse from the blood plasma into the interstitial fluid, you have a greater amount of release of oxygen from the erythrocytes according to the dissociation curve, allowing additional oxygen to diffuse to the tissues that need it. We also want to deal with carbon dioxide. Carbon dioxide is being produced via cellular respiration within these metabolically active tissues. As that carbon dioxide diffuses out of the cells from which it was formed, it will then move into the interstitial space. As it moves into the interstitial space, it will be at a higher partial pressure or higher concentration than in the blood in the capillaries, and as a result, it will have a tendency to diffuse from the interstitial fluid into those capillaries. Now again, we run into a second issue, that the blood can only hold so much carbon dioxide. The transport of carbon dioxide happens in a more complicated fashion than you might otherwise expect. Carbon dioxide from the respiring cells diffuses into the blood plasma, and then it gets into the erythrocytes, into those red blood cells. The carbon dioxide will then form carbonic acid, forming a hydrogen ion and a bicarbonate ion. That hydrogen ion can then actually bind to the hemoglobin itself. The red blood cell then has the ability to release the bicarbonate ion back into the blood plasma. This process naturally happens on its own, but it speeds up due to the activity of carbonic anhydrase. In addition, the presence of that hydrogen ion attached to the hemoglobin also allows the hemoglobin to bind to a carbon dioxide. As a result, as you have carbon dioxide moving into the erythrocyte, you have the activity of carbonic anhydrase, which will lead to the formation of bicarbonate ions and hydrogen ions. Those hydrogen ions will then bind to the hemoglobin, allowing additional amounts of carbon dioxide to bind on as well. Therefore, Transport of carbon dioxide by the blood happens in one of three main ways. The first of which is just as gaseous carbon dioxide, but that only ends up being 7% of the total carbon dioxide metabolized and transported in the blood. 23% of the carbon dioxide within the blood is bound to hemoglobin, and then 70% of the carbon dioxide being transported within the blood ends up being found as bicarbonate ions. Once you get to the lungs, that carbon dioxide in the gaseous form begins to diffuse out of the blood into the alveoli. This decrease in carbon dioxide then will shift the chemical equilibrium in favor of taking that bicarbonate and having it shift back into carbon dioxide and H2O. This is again assisted by carbonic anhydrase, which will lead to additional production of carbon dioxide from those bicarbonates. Finally, this reduction in the hydrogen ion concentration means that hydrogen ions will move off of the hemoglobin and also stimulate the release of the carbon dioxide from that hemoglobin, preparing that hemoglobin to store more oxygen. And that's us for right now. When we come back, we'll be talking about the final system for the semester. We'll be talking more about the kidneys. But till then, stay safe, stay healthy, and I'll see you later. So you got a new lecture topic for me? Yes, sir, I do. The avian respiratory system. Oh, birds are tight. What an odd thing to say. Yeah, forget I said that. So tell me about this respiratory system. Well, instead of breathing air into their lungs, they draw air through their trachea into a set of posterior sacs. Won't they have problems breathing with all that air in their posterior? No problem at all. Barely an inconvenience. They simply squeeze their posterior sacs, which pushes air into their lungs. So then they breathe the air out of their posterior? Yes, sir, they do. The air flows into tiny tubes called parabronchi and then into anterior air sacs. It's all very efficient. Wow, wow, wow. Oh, do they do that counter-current exchange thing where air flows in one direction and blood flows in the other? Well, they just use cross-current. But why do they do that? I don't know. Because that's the way it happens. Wouldn't counter-current exchange be much more efficient? Well, they just use cross-current. I don't understand. Look, sir, I'm going to have to ask you to get all the way off my back about this counter-current thing. Okay, let me get off that thing. Thanks. So what do you think? Well, it sounds great, but I don't know who would want to study birds for a living.